I am just trying to continue what I did yesterday on the lac operon. Little confusion was there. I am trying to give a solution for it. See, I just told you that the operator, promoter, all these things are a part of the I gene or called the regulatory gene, and they help in either switching on or switching off. This is the point I explained to you. I will explain further the whole thing here. Now the operator normally will be connected to a repressor. That means they will be binding together. When they bind together, the promoter, though it has polymerase and the other factors, promoter factors are there, it cannot move, or the repressor will not allow the entire promoter complex which is present here, required for the transcription, especially polymerase which reads the sequence, will not be able to move, and therefore that condition is called the off. That is when the repressor molecule, that is the protein molecule, attaches itself to operator, then it is not possible to go further to read the mRNA molecule for transcription. That is the position called off. Now when once a enzyme called ALU enzyme here, ALU transcriptase, when it comes and attaches to the repressor molecule here, it attaches, then the configuration of the molecule, that is protein molecule, will change. When it changes, it changes, the site of attachment will be changed completely. So it becomes different, and then there will be a gap that allows the molecule to move. See, we are all talking in terms of the spaces here only for understanding, but the space between them will be in the order of microns, or not even microns, in the angstrom units. That is 1/1000th of a meter. So 1/1000th of a meter. So that will be so small. So when once that enzyme attaches here, then the whole configuration of this will change, and this will give away, and then the entire promoter complex containing RNA polymerase, CAP, and the acetyl AMP, all they move in such a way, they start reading the mRNA, and then they copy ZYA, and then they produce the three important parts, that is galactolysis, permease, and transactylase. All these three are produced. All the three are important, because this helps, all these three will help in the metabolism of the glucose. So just remember this particular point. When the repressor molecule attaches itself to operator, transcription is not possible. When the repressor molecule gets attached to an enzyme, and that enzyme will change the configuration of the repressor molecule, and then it might become an inducer. When the enzyme attaches, it becomes an inducer and allows the entire complex, that is the promoter site complex, with RNA polymerase and the CAP and other things, will start moving, and they start encrypting, and ultimately transcription is possible. So this is a little point we have to remember. As I told you, this particular video what we have made is not for a higher level or for the medical students. It is only for the PUC students whose NCERT textbook has given very little information that is expanded. Thank you.